Hi everyone. Today's lecture is on glycogen metabolism. Glycogen is a highly branched glucose polymer for increased solubility and rapid degradation. Why store glucose as glycogen, not as fat? Fatty acids cannot be released from fat nearly as rapidly as glucose can be released from glycogen. Fat cannot be used as a source of energy in the absence of oxygen. Fat cannot be converted to glucose to maintain blood glucose levels required by the brain. Structure of glycogen. Glycogen consists of a branched polymer of glucose that is formed on a tyrosine side chain of the glycogenin protein. The glucose residues are mostly linked in alpha 1,4 fashion and occasionally in alpha 1,6 fashion to create branch points. Branching increases the solubility of glycogen. During glycogen synthesis and degradation, there are limits of particle size. The smallest particles contain about 2000 glucoside residues, the largest about 60,000. Small glycogen particles are also known as proglycogen, large ones macroglycogen. Glycogen in the liver. Glycogen particles are visible by electron microscopy after staining with a heavy metal, or by light microscopy after treatment with periodic acid shift stain, which generates a colored complex. Liver glycogen versus muscle glycogen. Muscle and liver store the largest amounts of glycogen. Most other cells store only a small amount of glycogen. The liver of a typical healthy postprandial adult contains up to about 100 grams of glycogen. It's about 7% of the wet weight of the liver. In the absence of exercise, the skeletal muscles contain up to about 400 grams of glycogen, or about 2% of the wet weight of muscle. If a person exercises to exhaustion and then consumes a meal very rich in carbohydrates, the exercised muscles can contain as much as 5% of their wet weight as glycogen. Physiological importance of glycogen. Liver glycogen is known as glucose reserve. It regulates blood glucose or maintains blood glucose homeostasis. Muscle glycogen is known as energy reserve. It provides energy for muscle to perform strenuous exercise. Approximately daily time course of the amount of glycogen in the liver of a resting volunteers. Glycogen in liver and muscles is synthesized chiefly during the first few hours after a meal. In the subsequent fasting period, when glucose juice exceeds glucose influx from the intestine, liver glycogen is degraded to glucose, which is released into the blood. This helps maintain a normal fasting concentration of glucose in the blood. Sources of blood glucose during a normal day. As shown in the figure, between meals, the blood glucose is derived primarily from hepatic glycogen. Depending on the frequency of snacking, glycogenolysis and gluconeogenesis may be more or less active during the day. Late in the night or in the early morning, after depletion of a major fraction of hepatic glycogen, gluconeogenesis becomes the primary source of blood glucose. The net direction of glucose flux in the liver via the cycle in the fat fasted and the normoglycemic conditions. In the normoglycemic conditions, there is no net flux of glucose. In the fasted conditions, the net flux in the direction of glucose release. In the fat condition, the net flux is in the direction of glucose uptake. This figure shows 5 stages of glucose homeostasis. These phases are phase 1, phase 2, phase 3, phase 4, and phase 5. The nutritional stages of phase 1 is known as well-fed condition. The origin of glucose is exogenous, Coming from diet, the tissue using glucose, almost all the tissues using the glucose, the major fuel of the brain is glucose. Then we move on to the phase 2. This is known as fast absorptive phase. The origin of blood glucose from hepatic glycogen and gluconeogenesis. All except liver, muscle, and adipose uses glucose at diminished rates. Major fuel of the brain is glucose. Phase 3 is known as early gluconeogenic. The origin of blood glucose is hepatic and renal gluconeogenesis. Tissues using glucose, brain and RBC, small amount by muscle. Major fuel of the brain is glucose and ketone bodies. The phase 4 is known as prolonged fasting. The origin of blood glucose is renal and hepatic gluconeogenesis. Tissues using glucose, brain at a diminished rate and RBC at normal rate. Major fuel of the brain is ketone bodies and glucose. Pathway of glycogenesis. The pathway of glycogenesis from glucose involves 4 steps. Conversion of glucose 6-phosphate into glucose 1-phosphate by phosphoglucomutase. Activation of glucose 1-phosphate to the sugar nucleotide uridine diphosphate glucose by the enzyme UDP glucose pyrophosphorylase. Transfer of glucose from UDP glucose to the glycogen in alpha-1,4 linkage by glycogen synthase, the major regulatory enzyme of glycogenesis. Glycogen branching enzyme, a trans-glycosylase, transfers some of the alpha-1,4 linked sugars to an alpha-1,6 branch. The major regulatory enzyme of glycogen synthesis is glycogen synthase. In muscle, glycogen synthase is activated by insulin, glucose 6-phosphate, glycogen depletion. In liver, it is activated by insulin, glucose 6-phosphate, and fructose. In muscle, the glycogen synthase is inhibited by epinephrine. In liver, it is inhibited by epinephrine and glucagon. Pathway of glycogenolysis. The pathway of glycogenolysis begins with removal of abundant external alpha-1,4 linked glucose residues in glycogen. This is accomplished by glycogen phosphorylase releases glucose from glycogen in the form of glucose 1-phosphate. The glucose 1-phosphate is isomerized by phosphoglucomutase to glucose 6-phosphate. In the liver, the glucose is released from glucose 6-phosphate by glucose 6-phosphatase enzyme, and the glucose exits via GLUT2 transporter into the blood. The rate-limiting regulatory step in glycogenolysis is catalyzed by phosphorylase, the first enzyme in the pathway. The major regulatory enzyme of glycogenolysis is glycogen phosphorylase. In muscle, calcium ions, AMP, epinephrine activates glycogen phosphorylase. This enzyme is activated by glucagon, epinephrine, norepinephrine, and ATP in the liver. Glycogen phosphorylase is inhibited by insulin, glucose, and fructose in the liver. Production of glucose by glucose 6-phosphatase. A glucose 6-phosphatase in the endoplasmic reticulum hydrolyzes glucose 6-phosphate to produce glucose. The hydrolysis requires 3 different activities. A glucose 6-phosphate phosphate antiporter in the membrane of the endoplasmic reticulum. Glucose 6-phosphatase activity that hydrolyzes glucose 6-phosphate to glucose plus phosphate. A glucose transporter that releases glucose from the endoplasmic reticulum into the cytosol.